Good morning, esteemed shareholders, potential investors, fellow board members, and distinguished guest, Mr. Raymond Rodriguez-Torres. My name is Rajan Ahuja, and I am the chairman of the board of directors. It's my pleasure to welcome you to Biomarck Shareholder Meeting and to sincerely thank you for taking the time to be in attendance this morning. As the chairman, it is an honor to open today's meeting, and I'd also like to acknowledge and thank my fellow board members. They are extremely valuable to Biomarck, and they work diligently and tirelessly on your behalf. Biomarck's board of directors have achieved many remarkable accomplishments and received numerous accolades for their impressive work. Some of these honors are being named the fastest growing company in the region, CEO of a clinical research organization, experience in obtaining FDA product approvals, as well as licensed major drugs to the top 3 pharmaceutical companies in the nation. 3 medical doctors and 2 PhDs occupy positions on the board. Biomarck's directors are comprised of a competent and dedicated team members who meet on a regular basis and are extremely committed to our mission and your investment. One individual board member I would like to acknowledge is my vice chair, Mr. Thomas Robert. Tom is a co-founder of Biomarck and has been a pillar of this company since its inception. Thank you for passing the chairmanship torch to me, Tom, and I humbly accept this honor and I hope to make you proud. Ladies and gentlemen, today you'll be hearing from 3 illustrious speakers, and I'd like to say a few words about each of them. We're so fortunate to have with us our special guest speaker, the chairman of the Live Like Bella Foundation, and my friend, Mr. Raymond Rodriguez-Torres. For the past few years, I've gotten to know Raymond, his family, and also his foundation. Sadly, my introduction to the Torres family came after their beautiful youngest daughter, Bella, passed. For 19 years, Raymond has enjoyed a successful career as an executive for large healthcare global organizations. He has managed sales of over $300 million and directly led a team of over 150 people. Raymond has worked with major pharmaceutical companies such as Sanofi Aventis, Hoffmann-La Roche, Genentech, and Therex Corporation. Raymond is the author of several best-selling books, and his book, Why Not Me?, which is a true story about a miracle in Miami, peaked at #1 on Amazon's top 100 hot new releases. Raymond has a passion for discovering a cure for pediatric cancer, and Biomarck is blessed to have Raymond's foundation as a strategic partner in our quest for fighting this insidious disease. Our second speaker, Dr. Ken Adler, many of you know from his years of Biomarck science and research and his perseverance in the cause of medical advancement. In my humble opinion, some of Doctor's greatest achievements to date will be presented here, and not only for the financial benefit of the investors, but more importantly for the greater good of mankind. It is my hope and purpose to facilitate Dr. Adler's mission, which is to make his work accessible to the sick and dying. I only ask that you support Dr. Adler, Biomarck, Raymond, and all of our efforts in this crucial effort. Driving and hosting this meeting is our third speaker, Biomarck's Chief Executive Officer, Dr. Brian Dixon. Dr. Dixon assumed the position of CEO in November of last year, and prior to that, he was Chief Medical Officer for Biomarck. Working with Brian over the past year has been an inspiration and a privilege. I am personally honored to have him represent Biomarck as our leader and look forward to Brian carrying our company to fruition. Ladies and gentlemen, I thank you for your time and consideration. I hope you are inspired and help us in this quest to potentially save lives and provide a better quality of life to those in need. Please take the time in welcoming my friend, Mr. Raymond Rodriguez.
Muy buenos días. Good morning. Thank you very much for welcoming me this morning, and I will say that it's a very distinct privilege for me to be here. I've come all the way from Miami not because I am an investor in Biomarck. I've come here because I believe that there is an opportunity here to help save generations of children and families battling cancer that is absolutely remarkable. I think it's worthy of all of our most careful consideration. And I'm uniquely qualified to speak on why I believe this, not only because of what my family and I have had to endure, as you'll learn a little bit more through the life and story of my daughter Bella, who passed away 30 months ago after a 6.5-year battle with pediatric cancer, but also because of what I've done professionally for the last 20 years in launching 13 orphan drugs throughout the history of my career. The great irony of my daughter's battle with cancer is that there was nothing to treat her with. And only a few months after her passing that I have the honor of meeting Ken Adler and learning about this remarkable science, which is the reason that drives me and fuels me to be here today and share this information with you. And so it really is an honor for me to be here on behalf of my family, on behalf of the board of directors of Live Like Bella, to come and visit and share with you why I believe so strongly in what this extraordinary company and this remarkable science can do for people. With that said, I'd like to give you just a little bit of flavor of what brings me here today and what Live Like Bella is all about. Ladies and gentlemen, I am a father who is a member of a club that no one would want to be part of. I held my little girl in her first breath on earth and in her very last. And as challenging as that sounds, and it is, Bella's life, as you could see from the video, was an extraordinary gift. I learned many, many things from my little girl. And perhaps the greatest lesson I learned is that it's not the weight of the load that breaks us, it's how we choose to carry it. What I said at the end of that video, which was recorded about 2 years ago, remained true then and remains true as I stand at this podium today. My goal is to make pediatric cancer a treatable and curable disease. And the reason why I'm here today is because I believe that this company has something that can bring us so very close to that goal. Bella's life impacted millions of people around the world, and Live Like Bella has since, in the 30 months that it has been in existence, has funded 9 clinical trials, have assisted families all around the world, and we do something that no one else does, and we pay for headstones and funerals when children unfortunately die of cancer. And that's very, very difficult to do, but I'm extraordinarily happy that Live Like Bella is uniquely prepared to do that, because these families face a situation after a long battle with cancer where they're in emotional distress, they're in financial distress, and every fiber of their being in that unit is stressed to a level that is frankly unimaginable. And so I'm privileged that Live Like Bella does that, but I'm here today because I'm so extraordinarily excited about what the future holds because of what Biomarck is doing. I think it's important to give you just a little bit of background of what you saw within that presentation. But pediatric cancer is the #1 disease killer of children. Every 3 minutes, somewhere in the world, a child is diagnosed, and in the United States alone, every single day, 46 children are diagnosed and 7 children die. Now, this may come as a shock to you, but unfortunately, we hear nothing of this. And I hate to use this comparison, but I will, unfortunately. If, God forbid, and I mean this truly, a gunman walked into a school and shot 46 children every single day, and every single day of those 46, 7 died, it would be an outrage. It would be on every news station. We would not cease to hear about this problem. And yet this is happening each day, not because of gunfire, but because of pediatric cancer. Over the last 30 years, with the exception of childhood lymphoma and childhood leukemia, we have not seen an improvement in survivability. And over the last 26 years, there have been only 3 drugs approved for pediatrics compared to hundreds for adults. Those words that went viral, Live Like Bella, which began shortly after LeBron James and Dwayne Wade wrote that on their shoes during the NBA Finals, mean a lot of different things to a lot of different people. For some, to live like Bella means to live in the moment. For some, it means to live courageously. But in its true essence, what Bella taught us that it meant was to serve others. That's what her greatest concern was. And that's what we have the opportunities to accelerate this and change some of these horrific statistics that we see today and make an impact on the #1 disease killer of children. In terms of the federal government, less than 4% of its federal funding goes specifically to pediatrics. And these are the reasons why we see the travesty that exists in the #1 disease killer of children. There has not been any advancement in this area because there's very little incentive. And although it is the #1 disease killer of children, it is a drop in the bucket statistically in comparison to the number of adults that have it. And so our goal in Live Like Bella has been not only to accelerate innovation at academic institutions, but work specifically with industry, and that industry that would have the courage and the wherewithal to specifically tackle this extraordinary problem, which is the reason why I'm so proud of the people that work for this company that have the courage to do that. So how does Live Like Bella address childhood cancer? Well, I think for those of us that have faced any significant problem in our life, which would include all of us, how do you eat an elephant? One bite at a time. You have to look at it from factorial aspects, and these are all the different things that Live Like Bella does. But one of them that we do uniquely is not only work with the academic institution, the public sector, government, and I'll talk a little bit about the Childhood Cancer Collaboration Act and something called the Bella Bill, which we've been advocating for, and I'm very happy to say that we have bipartisan support at a federal level regarding a piece of legislation that I believe can accelerate innovation for drug development, specifically in children, but also strategic partnerships such as these. And this is something that we're extraordinarily excited about. I think it's important for folks to understand how the funds are distributed at Live Like Bella, because we do 3 distinct things. We provide funding for research, we provide in-treatment support for children that currently have a child battling cancer, and lastly, memorial support. And you can see the distribution of these funds. We are undoubtedly a research-driven organization because we believe that that is absolutely critical. And so how have the funds been used? Live Like Bella is a relatively new organization. All of us have day jobs. But it's run by an incredible network of people that have said enough is enough. We need to do something about this, and I'm going to give of my time, my talent, and my treasure to tackle this. I personally am the CEO of the largest independent home health nursing company in Miami, Florida. No one takes a dime from Live Like Bella. And with that, I'm proud to say that with 2 years of 990 data to the IRS, 98% of all the funds that have been donated to Live Like Bella have gone to continue the charitable mission of what we're doing. And that's the reason why we've been able to impact so many families all across the world and accelerate 9 clinical trials in such a short period of time. And so these are the I would say the 24 months of significant progress that have happened. Here are some of the places where we funded trials. The first bullet point is actually incorrect. It's now 9 since it's been updated. Bella's Promise is an initiative specifically to work with the industry, which is what brings me here today. The Live Like Bella Pavilion is a building that's a 30,000 square foot building that we have been in negotiations with to build on the campus of Nicholas Children's Hospital in Miami, Florida, in partnership with the Ronald McDonald House Charities, so that families can stay for free while their children are receiving treatment, which is something that's very, very important. And as you can see, we've assisted hundreds of families all around the world, and we funded research both in academic institutions and with some biopharma partners as well. This was the latest announcement that we made. My daughter Bella was one of the first children from Miami to ever receive proton radiation. You see, when Bella was 4 years old, she became paralyzed overnight. There was no symptoms, there was no history. Our life changed in an instant. I was traveling. At the time I worked in pharmaceutical industry. I was at a meeting. I called home, and my wife answered with the terror in her voice that I had not heard before. And that was that throughout the night, Bella kept getting up and walking from the bedroom to this little Cinderella princess table that we had in the kitchen. And she kept falling. And by morning, she was completely paralyzed from the waist down. That's how this horrific life of ours in pediatric cancer began. And we found the reason and the cause for the paralysis. She had a main tumor around her spine, and she had metastatic disease in the jaw, the shoulder, the rib, the hip, the knee, the foot, the left hand, and the brain. And she was at diagnosis stage 4, group 4, alveolar rhabdomyosarcoma. And physicians that I've known my entire life told us, after scouring the earth, with no uncertainty, that my daughter would never walk again, and that she had a few weeks to a few months to live. And I can assure you that there is nothing that can prepare you for news such as that. But Bella didn't live a few weeks or a few months. She lived 6 years. She regained the ability to walk, run, and jump, and in the process inspired the world. And during the process of her treatment, in Jacksonville, Florida, at the University of Florida, she took proton radiation. At that time, in 2007, there was only 7 machines in the world, 5 in the United States and 2 out. Well, I'm happy to say that we announced at an event that we have every September called Bella's Ball, which is a very significant event where 1,000 people attend in person, and it's live transmitted to 15 different countries. Live Like Bella was able to fund the first proton radiation specifically for pediatrics in partnership with Baptist Health of South Florida at the Miami Cancer Institute, and this is a rendering of what that will look like. And this is the Live Like Bella Pediatric Proton Radiation Program, which is something I'm very, very excited about. But it brings me to impress specifically why I'm here, why I am so extraordinarily excited to be here and to share this information with you. And that has to do with the story of when I met Dr. Adler and Moet for the first time in Miami. You see, because of what I did for a living and the resources available to my family, we scoured the earth to try to find something to treat Bella with. We tested Bella's tumor all around the world, from the conventional to the borderline crazy. For those of us that are parents, you understand the desperation of trying to save your child's life. And we never found anything that worked. And lo and behold, we start Live Like Bella, and I'm introduced to a remarkable researcher who was doing research in COPD and bronchitis and found this amazing gene that, when abnormally amplified, caused cells to migrate very rapidly throughout the body. And as I started speaking to Dr. Adler and seeing some of the trial data, some of the histology, some of the videos of the mice that had been injected with triple-negative breast cancer, and at day 32, after being inhibited with this extraordinary advancement, found no tumors, I was shell-shocked with what I saw. And I went home, and I opened the document that I had in 3 years. And this was Bella's full genetic sequence. In 2010, Bella had a relapse in one of her ovaries. We were at Memorial Sloan Kettering Cancer Center in New York. Bella was treated everywhere. And in that process, when we removed her ovary, we worked with an organization in Baltimore to do a full genetic sequence of her tumor and to implant her tumor in xenograft mice to be able to run trials on mice that had Bella's specific tumor implanted in them. And what we received back from this very prestigious academic institution is a very voluminous document which has thousands of genetic data points. And on the first 2 pages of this, they do a very nice job of summarizing for you what they consider to be the clinically relevant or actionable gene mutations or gene amplifications. And unfortunately, as expected, there was nothing actionable. On page 27 of that report, after meeting Ken Adler and being so shell-shocked with what I saw, I found that Bella had the same gene that Ken Adler discovered here in North Carolina. And so I said to him, "Doctor, the most precious thing I have on earth is Bella's tumor. I'm going to send it to you." And we did. Now you have to understand something. When I say we tested Bella's tumor against everything, oh, we did. I personally ingested large amounts of scorpion venom. I personally ingested large amounts of rattlesnake venom. And I did so because if I was going to give it to my little girl, I was going to take it first. And we did that while we tested it in vitro in laboratories to see whether or not it would work. But the bottom line is that we were desperate, and the clock was ticking, and I would have done anything, and still would, to save my little girl's life. Every time, over the course of 6 years, we heard the words, "I'm sorry, stop what you're doing," it didn't work. After sending Bella's tumor to Dr. Adler, for the first time, I heard the words, "It worked." What you're seeing up in the upper left-hand quadrant here, under the title of alveolar rhabdomyosarcoma, is my daughter's tumor. What you're seeing there is day 0, and everything that you see in red there is rhabdomyosarcoma, the tumor that took her life. On the right side of that, what you see is after day 3. This is by far the most remarkable thing I have ever seen in cancer. From the laboratory at Miami Children's Hospital, we additionally sent neuroblastoma, which is the most common childhood tumor. What you see in the bottom left-hand quadrant is day 0. To the right of that is day 6. I don't know that I'm properly expressing to you, and I don't think that there's proper human words to be able to express and implore upon you the significance of this, not only for my daughter and what it means to countless numbers of children battling rhabdomyosarcoma and neuroblastoma, but also other tumor types that have been tested. I'm sure Dr. Adler will share with you later some of the work in canines, in dogs, in osteosarcoma. Osteosarcoma, bone cancer in children, is a horrendous cancer where the frontline treatment has to do with amputating the leg. And there's good reason for that. But even still, I have attended the funeral of more children with one leg than I would want anyone to ever attend. Why? Because they relapse. And this is the best we have to offer our children, which is the future generations of humanity. The time has come to change that. This is what drives me each and every day, in memory of my daughter and to benefit generations of children. And this, I believe, is the future. When you can think about taking cancer treatment and turning it into a chronic treatment, what better and who better than children for this? I mentioned the Bella bill, and one of the things that we're talking about in Washington and having a conversation is this. When an adult dies of cancer, unfortunately, the average loss in years is 20. When a child dies of cancer, the average loss in years is 67, almost 70 years. Let's develop a chronic treatment to keep people's metastatic cancer at bay. And we have folks taking this medication for almost 70 years. And I will tell you honestly that if I could have Bella here with me, taking 006 each and every day, I would do it every day and twice on Sunday. This is what makes me so excited about coming here. This is by far the most remarkable thing I've seen. And ladies and gentlemen, I've seen a lot of things in cancer. And I have funded a lot of research in cancer. And I worked in biotech and cancer for so many years. And this is the most exciting thing that I've seen. And so it's for that reason that I've come here today, to share this story with you, to hopefully inspire you. Live Like Bella will indeed be participating with Biomarck to accelerate this and do all that we can to fundraise and provide grants and provide avenues and opportunities for the appropriate children and adults to be able to take this at some point in the future and help generations of children and families battling cancer. And so in closing, I'll say thank you. Thank you for investing in Biomarck, for believing in the company, for coming here today and listening to my story and to Bella's story. Today is a day that we have the opportunity to look forward to help generations of people battling cancer, and specifically children. And for me, it's a great privilege to be here and share this with you today. I'll stop now, and I'll be happy to take any questions that you might have, either about Bella, about Live Like Bella, or anything at all. So the bill we are negotiating still is called the Childhood Cancer Collaboration Act. And if you're interested, I have some information I can give you afterward. But the best way that I can describe the Bella bill to you is as follows. So let's say, God forbid, any of the adults here had a pain that we knew wasn't just right. We wouldn't wait too long to go see the doctor because we're adults. We know we're susceptible to cancer. We've likely had someone in our family affected by a disease. Children don't do that. And so Johnny has leg pains for 8 months. And they go to the pediatrician. All blood counts are normal. There's no pinpointed reason why this is existing. And so the pediatrician, and I can say this because both my father and my older brother are pediatricians, more than likely will say, more than likely it's due to growing pains, and statistically they wouldn't be wrong to say that. Come back and see me in 3 months. Susie shows up with headaches. Same thing. Blood counts are normal, no pinpointed reason. Perhaps you're having migraine clusters. Perhaps you need to go see the ophthalmologist because the chalkboard is bothering your eyes. In the meantime, there's a roaring medulloblastoma growing there. Children have a physiological disadvantage to adults. They're growing, so their cells are rapidly dividing. So what do we know happens when you add a malignancy to an organism that's rapidly dividing? It's far more aggressive. And what we now know is that after every differential diagnosis fails, and eventually they screen the child for cancer, universally, children present stage 3 or stage 4. And so for that reason, and based on some of the data that I just showed, children are universally excluded from entrance into clinical trials because they are a statistical threat to the outcomes of the study. What we're calling for in the Bella bill is that pediatric data be excluded from the proposed adult indication of which the study is aimed for. And so therefore, if the children die in a clinical trial, either because they present with too far advanced disease on the outset, or it simply doesn't work, we don't want to hinder the advancement of breast cancer, prostate, lung, and others that have much larger populations. But at least children will get a shot at innovation in real time. That's the Bella bill. Thank you very much for having me. And I believe we have Dr. Adler next. Excellent. Thank you.
I will repeat the question. Well, good morning, everybody. I'd seen Raymond talk before, and I spent a lot of time talking with Raymond about Bella, about what we're doing, and how excited we are to be working with Raymond. And as he said, hopefully what we're doing here will have the kinds of results that we're anticipating, that is turning cancer into something chronic that can be treated. So I'm going to talk about the science. I'm sorry that I have to talk about science. It's always difficult to speak to groups that aren't as fully learned in science as some of my students. Well, actually, some of my students probably would fit right in. And I'm going to try to do is present this as clearly as possible. The work that's going on in my laboratory now and in the past few years has been truly remarkable. I'll give you a little bit of an introduction into who I am and how I got into cancer research. So I got my PhD from the University of Vermont College of Medicine in the late 1970s. I stayed on at the University of Vermont for several years as a faculty member at the medical school there. I came to North Carolina State University in 1987. I was recruited to run a graduate program in cell biology way back then. In 1998, I was named the Alumni Distinguished Graduate Professor at North Carolina State University. I've published a lot in journals. I'm the editor of the American Thoracic Society Basic Science Journal. Been all over the world speaking. My wife Pamela is here. She accompanied me to many of these venues. And I was just a basic researcher 15 years ago, not interested in developing drugs, just like the great majority of bench researchers. And we made this discovery in my laboratory in about the year 1999, 2000. And through the Tech Transfer Office at North Carolina State University, Biomarck was formed. I think it started in 2002 with the help of Tom Roberg and Fred Gant. Fred's wife, Beverly, is here attending this. So when I go around giving these talks or speaking about my science, I always say I'm one of the lucky researchers because the work that I did on the bench has been translated into an actual drug to treat people. So I want to talk today about some of the science behind the drug. And the first thing I want to talk about is the work we've done with cancer. And then I'm going to move to another disease called acute respiratory distress syndrome, abbreviated ARDS. So let's talk about cancer first. And I'll make this statement. The Bio-11006, the drug that is the lead drug for Biomarck, inhibits migration of cancer cells. These are cancer cells you put into a chamber in the laboratory, you treat them with Bio-11006, and the cells, which normally migrate like crazy, stop migrating, treating with this. Now, what does migration of cancer cells in a chamber mean? That can be extrapolated to metastasis of cancer cells in the body. And some of these cells that we work with, most of them have been adult lung cancer cells. We've done work with breast cancer cells. As Raymond pointed out in the slides that he showed you, was work that we had done looking at two childhood cancers, neuroblastoma and rhabdomyosarcoma. The rhabdomyosarcoma is what Bella died of. If you look at the literature, there is nothing that's been shown effective. This is the first time, to my knowledge, something's been shown effective even in a laboratory setting. And the last thing, as Raymond also mentioned, osteosarcoma, bone cancer. We've done work with canine osteosarcoma, which is very similar to human, as well as human cells. It stops migration of those cells in vitro, in chambers. Okay, and I think this is actually the mother slide of the one that you showed. This is rhabdomyosarcoma, and I'll just show you how effective this drug is, and I'll just walk you through this. So these are cells on a chamber. This is what they look like when they migrate from one chamber to the other. So this is where they've migrated to. You just count the numbers of cells. Actually, Yin Chi from my laboratory is back here, and Sue Fang, and they're the ones that actually count the cells. So this is what it looks like. This is what cancer cells look like when they move. If you would take normal cells and look at them, not cancer cells, you'd be hard-pressed to find any cells there. So cancer cells move. They metastasize, they move like very rapidly in chambers. So we treat these cells with different drugs. The first one is called RNS. That's just a control. Before you treat it with 006, you not just say, a good scientist would say, well, maybe if you just throw anything in there, it'll stop them from migrating. So let's throw a control peptide similar to 006 in, and it had no effect. And then with 11006 and increasing concentrations, you see fewer and fewer cells migrating. When you get to this, at the highest concentration, very, very few cells. It's like a 90% inhibition of migration in chambers. Okay, that's great, but what about looking at some sort of cancer in an animal? So we use mice in the laboratory, and mice do not get cancer. So when you hear people saying, well, we can cure cancer in mice, the truth is you can't cure cancer in mice. You have to give the mice cancer first, then you can cure it. So what we do is give mice lung cancer. And the best lung cancer models in mice is when you take human cancer cells, you inject them into the lung of a mouse, and sure enough, that mouse gets lung cancer similar to what a human gets. The tumors grow, the tumors metastasize to all different organs, diaphragm, to the spleen, to the liver. It's almost identical to what you see in humans. So it's called a model of disease. I think I have the word model in there somewhere, or one coming up. Yeah. So you're actually giving a mouse a human disease. And when we give these mice 006, either at the beginning or well into the course of the disease, it stops metastasis cold. And it's given to these mice as an inhaled aerosol. Now, we don't give mice a little thing to put on their nose and breathe in. We put them in a box in a chamber, and we put the aerosolized drug, and they breathe that. And sure enough, not only does it block metastasis in these mice, but if you combine it with the standard chemotherapy, we use cisplatin here. It's a platinum-based drug. Anyone who's had or gone through cancer or know people who have, they get things like cisplatin, carboplatin, Taxol. They all have the same effect. And when you combine that with 006, you get what's called a synergistic effect. That is that primary tumor shrinks, the metastasis stops more so than either given separately, or if you'd sum them, it's like a geometric increase in effectiveness. So I'm going to show you some data. Originally, when I prepared this talk, I had lots and lots of data, but they told me don't show too much data. So this is from a mouse. And what we did was inject this very aggressive human lung cancer into the lobe of the mouse. The mouse developed metastatic lung cancer. And when we looked at the development of these tumors, we found that the drug had a tremendous effect in blocking metastasis, and combined with the drug cisplatin, even more so. So let me walk you through this. So these are looking at the metastasis in the lung, left lung, right lung, always left lung, right lung. This is an untreated mouse treated with 25 micromolar of 006, increasing concentration, fewer and fewer tumors. If we combine it with cisplatin, this is cisplatin by itself, numbers of tumors. If we combine it with cisplatin, we again get a dose-dependent response. And look down here, the highest concentration of 006 with cisplatin, it's almost no tumors metastasizing. Let's look at some distal organs. That was the lung. Now if we look at metastasis to the diaphragm, sternum, ribs, and mediastinum, the same thing applies. Drug by itself, drug with cisplatin, cisplatin by itself. And here's the untreated mouse. And again, call your attention to this. Highest concentration with cisplatin, negligible metastasis. And I'll get into the mechanisms of how this works. One more thing, that was a model where we injected the human lung cancer cells into the mouse lung. The second model is you inject human lung cancer cells into the mouse tail vein. Again, it's not as easy as it might sound, but we have experts doing this. And you can inject these human lung cancer cells into the vein of a mouse. They will move to the lung, they will seed and develop tumors, metastatic tumors in the lung. It's a second way to do it. Rather than injecting directly into the lung, this is another model, and sure enough, the same sort of thing. So this is the untreated numbers of tumors at the end of the study, and these 006 and parent peptide of 006 mans. So it's working. It's blocking metastasis in two different models. I would point out, if you go into the literature, you look at some other studies that have been done, nothing stops metastasis like this. Nothing. Okay, I'm going to talk about the mechanism of action, MOA. If you hear pharmaceutical people say MOA, that means mechanism of action. We know the drug works. We see it blocking metastasis. How does it work? And I'm going to try to explain that to you and be as clear as I can. So before we talk about how it works, let me introduce you to three key molecules that are involved in the mechanism of action. And they're all interrelated, and I hope I'll be able to explain how they're interrelated. The first is Mark's protein. Mark's stands for, I'm sorry, I'm always pointing to my left here. I should be switching off between the left and the right. Mark's stands for myristoylated alanine-rich C kinase substrate. Mark's is the mark in Biomarck. Okay, Biomarck is founded and based on the studies that I did in my laboratory related to Mark's protein. Mark's, I just will point out, is a it's a molecule that's involved in movement. It's involved in movement of different molecules within cells, and it's also critical to movement of cells. Okay. I will also point out that Mark's is what's called an evolutionarily conserved protein. So if you take the Mark's molecule from an earthworm, a slime mold, and compare it to Mark's taken from a human, it's almost identical in three specific regions, functional regions of Mark's. So whenever you see a molecule that's so well conserved through evolution, that means that whatever it's doing well. There's no need to change it. So it's in all cells, all organisms, and whatever it does well. Second one I want to talk about, and I'm sorry for these acronyms here, is called PIP2, and it stands for phosphatidylinositol 4,5-biphosphate. PIP2 is a molecule which is critical to cell movement. And let me draw an analogy for you. So we've all seen these chain reaction collisions in the highway. So one car crashes into a next car, next car, next car, next car, next car. 10 cars, 20 cars, as many as 70 cars sometimes. Okay, so let's envision the first car being PIP2 and the last car being the cell that has to move. So under normal conditions, PIP2 will initiate a number of different reactions in the cell and will end up by hitting the first car. It will in turn activate a number of different molecules and end up with the cell moving. Okay? So PIP2 is critical for movement of cells. Interestingly enough, I said this is all interrelated. In normal cells, PIP2 can't stimulate the cell to move. And it can't stimulate the cell to move because Mark's binds to it and sequesters it. It doesn't allow PIP2 to initiate this sequence of events. So picture, if you will, a car, PIP2 car, and Mark's is the chock. We had to look up that word. Thanks, Robin. A wedge, a wooden wedge you put under the tire. So PIP2 it's ready to initiate this reaction, but it can't because that Mark's chock is blocking it from moving. Okay, so under normal cells, that's how it works. Now, when a cell gets stimulated, because sometimes you need a cell to move, if you have an injury and you need to get inflammatory cells to it, there'll be a signal sent to that cell that will cause marks to be activated, and that will cause the marks holding of PIP2 to cease. PIP2 can then stimulate and cause the cell to move. However, we don't want, under normal conditions, cells to move all the time. So what keeps this PIP2 from constantly causing cells to move? And the answer is a third molecule, and that's called PP2A. And again, I apologize for the acronyms. PP2A stands for a protein phosphatase. And protein phosphatase is the regulator. It puts the wedge, the chalk, back under the wheels under normal, in normal cells to keep the cell from moving. So it does what it's supposed to do. It stops. It's a nice endogenous, within the cell regulator of the cell function. Okay, so those are the three molecules I'm going to talk about. Before I do that, let me talk about some basic definitions in science. So when we talk about molecules being activated, Marks being activated, PIP2 being activated, PP2A being activated, the way that molecules are activated in biology is that they're phosphorylated. A phosphate group, which is a phosphorus and 4 oxygens, are added to the molecule. I won't go into all the details, but activation, phosphorylation, same thing. In return, deactivation, the phosphate group is removed, so deactivation is dephosphorylation. So when I talk about phosphorylation and dephosphorylation, I'm talking about activation and deactivation of a molecule. Okay. So, in cancer cells, what happens? Why are they different from normal cells? Well, Marks is highly phosphorylated. Okay, it is highly activated in cancer cells. And the way that Bio11006 works is it deactivates Marks, dephosphorylates it. Okay, let me talk a little about the mechanism here. So, I talked about PIP2 and how it initiates that process. And I hope I'm making this clear. So when PIP2 gets activated, when Marks gets activated, it releases its hold on PIP2, takes the chalk out from the wheel of PIP2. PIP2 starts hitting cars, and cells move. When cancer cells move, that's metastasis. Now, normal cells, as I said before, Marks is that chalk under the wheel. It keeps PIP2 from starting this process. Okay, keeps it from being activated. When Marks gets activated, phosphorylated, it releases PIP2, allows it to hit those cars and start the process, ending in cell movement, cell migration, metastasis. It is known, in fact, we published a paper showing this, that in cancer cells, Marks is hyperphosphorylated. So rather than having a nice, simple mechanism of Marks letting PIP2 go, then coming back in, Marks is too activated, too phosphorylated, that chalk never gets back under the wheels in cancer. PIP2 is constantly hitting those cars, those cells are moving. This brings into play PP2A, the third molecule. Okay, under normal conditions, the way that that chalk gets back under the wheel of the PIP2 car is that protein phosphatase 2A, PP2A, will dephosphorylate, deactivate Marks, allowing it to get back under the wheel. Okay, this is in normal cells. But in cancer cells, lo and behold, PP2A is inhibited. If If you go to the literature and do a search on a site called PubMed, which you can bring up any literature papers, you'll find maybe between 50 and 100 papers explaining that one of the biggest mutations in lung cancer, and other cancers even, is the fact that this PP2A is mutated. There is a protein called CIP2A. It's called cancerous inhibitor of protein phosphatase type 2A, which is the third most common mutation in lung cancer. I'm sorry, it's the third most common molecule found in lung cancer. So PP2A is depressed in cancer. Think about it now. So you've got that PIP2 car constantly hitting the other cars, setting into motion this sequence of events. The cells move, and it's all regulated by Marx. Marx normally keeps PIP2 from doing that, but in cancer, since Marx is hyperphosphorylated, it can't do that, and the cells constantly move. And the reason that it can't do that is because it can't be deactivated because PIP2 is repressed, it's inhibited. And because it's inhibited, as I said, you get excess Marx activation phosphorylation. Okay, let's go back to some data. It's nice to talk about this. What's my evidence? And by the way, what I'm saying here is totally new from my laboratory. This has never been published. In fact, it's unknown. Just digress for a second. I had published in 2002 a paper showing that Marx protein is dephosphorylated by PP2A. Okay? If I go to the literature now, the PubMed, and say, let me put these two terms in, PP2A, Marx, I think there are 3 papers, mine and a couple of others. I don't think they're in English. So this is really unknown. So if you go and you see, yeah, there's Marx phosphorylation in cancer, and yeah, there's PP2A inhibition in cancer, but putting them together as far as dephosphorylating Marx, and Marx being the primary target, is something that only my laboratory has been working on. Anyway, so what's my evidence for this? We can do in my laboratory, and we have 2 experts here who do it, you can actually treat cells with a molecular called a construct which silences the expression of any gene that you want, any protein. So normally, say you have an activity level of 20 PP2A in a cell. If you treat it with an siRNA to silence it, it goes down to 2 or 3. So you can really repress the expression of this particular protein. So when we treat cells, these are very, very aggressive human lung cancer cells, PC9 cells, the same ones that we inject into mice. And you treat them by silencing, repressing the activity of PP2A, the expression, we get a 2 to 3-fold increase in migration, which correlates with an increase in Marx phosphorylation. It's the first time I've ever shown anybody these data. So another thing you can do is buy a drug which specifically activates PP2A. That drug is called FTY-720. And what do you think would happen if you activate PP2A in a cancer cell? It would cause dephosphorylation of Marx. And when Marx gets dephosphorylated, you get less migration. Sure enough, dose-dependent manner, we can block migration of these cells by activating PP2A. Now, we know that Bio-11006 blocks metastasis. All these molecules I'm talking about, how does it work? Well, it turns out that Bio-11006 acts to deactivate Marx, dephosphorylate Marx, allowing it to get back into underneath that wheel of the PIP2 car by actually activating PP2A. Again, this has never before been shown to anybody, so it's exciting for me. And here are the data showing the activity of PP2A using the FTY, the drug that you can buy to activate PP2A, 006, works even better than that in a dose-dependent manner. So that's how it works. That's how it dephosphorylates marks. That's how it blocks metastasis. Another thing about cancer. We talked about the synergy between 006 and cisplatin or any of the platinum-based chemotherapy agents. Well, the chemotherapy now is based on, as I'm sure Raymond knows well, cancer cells divide rapidly. So the drugs that are used, the chemotherapy drugs, will kill cells that are dividing rapidly. Cancer cells, unfortunately, when they give them to humans, other cells like hair follicles also get affected. And the reason that they work, or cells that are dividing rapidly, I don't want to get into too much, get too deep into this, but cells, when they divide, the chromosomes line up, the cells pinch off, and two daughter cells come from a single cell. A lot of movement, okay? And one part of that phase, when the cells are dividing, is called G2M. It's just different acronyms for different parts of the cell cycle. So G2M is is when cells are actively dividing, okay? All these drugs, cisplatin, Taxol, carboplatin, will kill cells that are in G2M. That's their target. So if you can get cells to stay in G2M, that would be an advance in cancer therapy. Guess what? 006, by affecting Marks, again, Marks is involved in movement. So 006 blocks Marks activation. It keeps cells from dividing, keeps them in that G2M phase for long periods of time, called G2M arrest, and then makes them much more susceptible to these chemotherapeutic drugs. This is like almost an added bonus to the effects of 006 in cancer. So I presented all the latest work from my laboratory on how it works and some of the data related to how effective this is in cancer. Let me switch gears now to a second disease called ARDS. I talked before about ARDS, acute respiratory distress syndrome. It's caused by a variety of different stimuli. The most common are patients with sepsis, bacterial or viral pneumonia, trauma, smoke inhalation, near drowning, etc. The firefighters who inhale smoke actually suffer from ARDS. And what causes ARDS? The primary cause is you get all these inflammatory cells rushing to the lung, releasing all these bad mediators. Okay, movement, cell movement, what does that involve? Marks. So in the United States, there's more than, I think it's close to 250,000 cases a year now. I think it was 240,000. And about 40 to 50% of these patients will die within a month. It's a very lethal disease. And unfortunately, there are no drugs available. Similar to, I guess, stage 4 cancer, there are no drugs available to treat ARDS. The only way they're treated with different ways of giving them oxygen. Okay, so what we did in the laboratory was try to recreate ARDS in a mouse. And the model, again, to recreate the human disease in a mouse, you give them the outer coat of bacteria, lipopolysaccharide, abbreviated as LPS. You give that in their lungs, and they get essentially a disease just like human ARDS. And lo and behold, when we give the mice 006, it actually cures the disease. Not only does it prevent it if you give it before, but it cures the mice. Again, never before been shown that you can actually reverse this disease process in an animal. And I think some of you have already seen the video of these mice. But those of you who haven't, I'll just show them. And hopefully it will work. Can get the lights dimmed, Ted? So this is a mouse. These are mice treated only with LPS 48 hours afterwards. These mice are sick. You can see their hair standing up. You can see their eyelids drooping. They're lying, hanging out in their own urine, which you never see. Look at their hair standing up. Hiding in a corner, labored breathing, all sorts of bad things. Now, when we look at the same mice, or same group of mice, treated starting at 24 hours after they got the LPS. It says MANS on here, but it's actually 006. This white stuff here is just cotton you put in to keep the mice happy, and you can see the difference. Their coats are clear, their eyes are bright, they're moving, they're trying to get out of the cage. They've essentially been cured of the disease. All right, we talk about method of action. What about the method of action here? Well, just like Bio-11-006 blocks metastasis, it also blocks the influx of inflammatory cells into the lung, given as an aerosol. And this is actual data from the laboratory looking at inflammatory cells, neutrophils over here. Somehow the label got lost here. So now we're looking at numbers of neutrophils in the lung. What we're doing is starting the treatment with 006 at 0 time, right when you give them the LPS, 4 hours later, 12 hours later, 24, 36 hours later. So no treatment, no LPS. Mice have very, very few of these inflammatory cells, these neutrophils. This is the primary inflammatory cell. They're white blood cells. After you give them LPS and you look at 72 hours, enormous 80-fold increase in these cells. And look at the beautiful straight line we have here. 0 time, 4 hours, 12, 24, 36. The mice you saw in the video were treated here at 24 hours after it. So they have large numbers of neutrophils, but there are no more neutrophils coming in. It stops the neutrophil influx cold. Again, something that's never been reported before. So I'll conclude by saying that 006 could become the first drug effective in diseases where there are no drugs. And there are very, very few therapeutic options. Our work is based on lung cancer. And Brian's going to talk to you, Dr. Dixon's going to talk to you about clinical trials based on lung cancer. However, with all these other studies that I've done in the laboratory with breast cancer, rhabdomyosarcoma, neuroblastoma, osteosarcoma, there is no reason to think this could not be effective against other cancers. And work in my laboratory for the next few years is going to be based on looking at potential of this drug for use in all kinds of cancers that metastasize. And of course, Brian will talk to you also about trials that we have in ARDS, another disease which there is no drug and basically there is no cure. So I'll stop here. Thanks very much for your attention and be glad to answer any questions. Walker.
Ken, do you know why the EDA2 is inhibited in cancer and how the reversal is accomplished by 11006?
As far as why it's,
What does it? What's the mechanism in cancer?
as I said, there are mutations in PP2A, which are very, very common in cancer. There is expression of several different molecules, which are actually inhibitors of PP2A. It's one called CIP2YA, cancerous inhibitor of protein phosphatase type 2A. There's another one called SET, which is also acts by inhibiting PP2A. The work that I've focused on is in lung cancer. There are trials, there are studies going on in prostate cancer where it's also very much inhibited. So the mechanism could be mutation in PP2A, or it could be expression of proteins which inhibit PP2A. I would point out that PP2A has a number of other targets other than Marks. So a lot of the work in the cancer field related to PP2A has been looking at other targets of PP2A. The work related to Marks is only recently, as I said, if you look at Marks and PP2A, there'll only be a couple of papers and mostly the stuff from my lab. Your second question is how it works. I can tell you what I think, as this work that's going on in the lab now to try to figure this out. So under normal conditions, we know that 006 is actually a part of the Marks molecule. It's it's one end of the Marks molecule. And under normal conditions, that part of the Marks molecule is inserted into the cell membrane. It never gets to the cytoplasm where PP2A is. So I think that when Marks gets activated, phosphorylated, in normal cells, it comes off the membrane into the cytoplasm, and PP2A, via some binding or some sort of recognition factor or some potentially other proteins, is able to recognize that Marks has come off the membrane. It's time to put it back on the membrane by dephosphorylating it. I think the signal for that is that part of the Marks molecule, which is normally cryptically hidden from PP2A. We're doing studies in the laboratory now to show that 006, or the Marks, that part of the Marks molecule, will activate PP2A by some sort of binding or recognition. Okay, sir.
Two questions. You talk about how you inhale 006, and that's so effective. How would this drug be delivered to other forms of cancer, the breast, other things? And number two, how dangerous is it to work in your lab?
I'll answer your second question first. You'd have to talk to my people here, but it's actually a very nice environment to work in my lab. It's not dangerous. None of the drugs that we work with, like 006, has been shown to be totally safe in humans. It's been tested in human patients already.
I'm talking about the aggressive cancer cells, which you might get.
Oh, no. Now, these are, you can't get that from that. And the bacteria stuff, you have special hoods and safe procedures to work with bacteria and viruses. You don't even do it in my lab, you do it in another place. Okay. Isn't that true? The fact that you, okay. You heard it from Yinchi's mouth. Your first question was, yeah. Yeah. Delivery of drugs to solid tumors. Let's take pancreatic cancer. Okay, one of the problems in treating that is you can't get a drug to that specific target. There are numerous mechanisms in play or ways to deliver drugs to targets now that are subject of very, very rapidly growing and fast-changing discoveries in targeting drugs to specific areas. One simplest thing that I can think of, actually, is someone at NCH, I believe, who's doing this, is you can use a nanoparticle. I think we all have heard or seen nanoparticles. So these are actually iron. You can make a nanoparticle out of something like iron, put the drug inside it, use a magnet, inject it into the bloodstream, use a magnet to actually draw those nanoparticles to your organ of choice, and you can visualize it. The imaging now is so precise, you can actually visualize that by actually do it in real time and visualize it. You can put an antibody against a tumor. Tumors have specific markers, antigens on their surface, only for tumors, on specific types of tumors. You can put an antibody against that tumor antigen on the outside of a nanoparticle that has the drug. It will be drawn to that antigen and release its cargo when it comes into contact with that. As far as inhaled, something like breast cancer, I don't know if Once it's inhaled, it changes into another form of 006, which is just as effective. So how long that persists, whether it would also affect breast cancer, I don't know. We are doing some studies, planning some studies in mice. So there are other ways to deliver the drug if it's shown effective. But I don't have the expertise to do that. We'd have to work with other people that spend their lives doing this. I know at NC State they have a big nanoparticle facility, and in fact, we are considering working with some of those people to develop a way to do that. Okay, thanks very much for your attention. And I guess I should introduce Brian Dixon, the CEO of Biomarck.
Thanks, Ken. As Rajin kindly said in his introduction, I am the CEO now, and I welcome you all here. I particularly want to thank Raymond for coming up from Miami. He is right. Childhood cancer is very difficult to treat, and there is very little research. His video is heart-wrenching, but it's also inspiring. There's no doubt about it. No matter how many times I see him give that presentation, it's hard not to be moved. I'd also like to thank Ken, not just for introducing me, for three things. One, he was the inventor and the work around the Marx protein. And that can't be understated and shouldn't. I'd like to thank him for his scientific work, which is obviously outstanding. And then lastly, I'd like to thank him for his understanding, because I cut his slides in half. So he originally came with double those slides in a true academic sense. And so I thank you for your understanding on that. People traditionally put up their resumes. I'm only going to highlight one thing. I've led teams that have submitted successfully, in other words, had approved 17 NDAs and made many presentations to FDA advisory boards. When designing clinical studies, and clearly we should go into the clinic again. I'll mention why I say again, because obviously this has been into the clinic for COPD indication. But when designing studies, it's really important to get it right the first time. Clinical studies are not cheap, and they take time. So it's money and it's opportunity in terms of time. So you don't design them in a vacuum, no matter how good you think you are. You take advice, and we have taken advice. So I'm listing here some people: UNC Lineberger, Scott Gottlieb from the FDA, Rosenblum from Johns Hopkins, Neil Reedy from Duke, Michael Mathey, who's world famous in terms of ARDS treatment, and he's used by the FDA as a consultant for that, and then Shannon Carson at UNC. So I've met or read through or spoken to all of these people to try and make sure that when we do a study, we do it right. Because it's not only what indication, it's what type of patient, what dose, how often are you going to give it? Are you going to give it by a nebulizer or IV or intraperitoneal? By the way, 006 has been given IV and intraperitoneal in animals, and it worked just the same. So, but can you maximize the formulation? And in mentioning that, I just want to acknowledge two other people. Dr. Walker Long has also given, he should be on this list because he's given me a lot of advice, frequently. But I welcome it, I really do. He's an investor and he's also on the board. And he's had a lot of experience in clinical studies. The other person I want to recognize, though, is Dr. Indu Parikh. Indu sitting here. He's our chief scientific officer, and he invented Bio 11006, the formulation and its backup, 10901. So what a tandem they are. One invents the relevance of Mark's protein, and the other one goes through and gets our formulations and our peptides. Not just those two, we've got a stable of over 100, and that's important. I'll come back to that. By the way, just to make sure we all understand, because we're talking about proteins, in this case, Mark's protein, and peptides. Well, a protein is a long chain of amino acids strung together. So in Mark's cans, it's over 300. It's 332. So it's a long chain of amino acids. That's a protein. A peptide is a very short chain. In the case of 11006, it's only 10 amino acids. So we're using the short one to control the big one. That's really what it boils down to in the simplest sense. So a peptide is a very short chain of what a protein is. All right, so I took all this opinion, and I just want to review, having asked Ken to take out half of his slides, I'm going to refer to them again, but it's from a different point of view. All right, he looks at them and gets excited from an academic point of view. I look at his data, and we discuss every week his data. But I'm looking at it from a different point of view. It's a clinical point of view. How relevant is this to a treating clinician, to the FDA, or to a company that may want to co-develop with us or acquire us, for example? So what would I distill of the relevance of his work? Well, the first is it's an effect in many cancer cells. So yes, we're going to choose lung cancer, and that's where the bulk of his work has been focused on lung cancer. But to an acquiring company, it's nice to know, well, if we threw money at this thing, we could do rhabdomyosarcoma, neuroblastoma, breast cancer, etc. We may want to optimize the formulation because Indu picked this formulation with good reason for COPD. And we can use it with good reason for lung cancer, it's in the lung, and for ARDS, also in the lung. But if we went down other cancers, it's a very good question, we would look to optimize or change that formulation. But right now, we're happy with what we've got. Because modern nebulizers are handheld, and inhaling this product twice a day, even if it's over a long period, you can do it at home. Whereas for cancer drugs, you have to go to a hospital, and they have to be injected, and you have to go repeatedly. So it's actually a bonus in the sense that it's a nebulized form for the moment. He's shown dose response in many of his slides. That is really important. If you don't get a dose response, people at the FDA, for example, start wondering, was that just a lucky shot? Okay, this is the number, and then you treat them, and it comes down to here. That may have been, but if you show a dose response, in other words, the more of the drug you give, the better result you get, that's not by chance. So dose response is something that the FDA look for, and it's what an acquiring or partnering company, Pfizer, for example, would look for. The method of action, a lot of these slides were about that. A lot of work has been done in the last year, and I'd like you to publish it, by the way. We have patented it, by the way, included in our patents. But method of action is really important, again, for people from the outside looking at why, okay, your drug works, but why does it work? And that's what a lot of Ken was focused on. The animal models he was talking about, they're not just taken by chance. They've been shown in the past to be the most predictive animal models for an effect in humans. Doesn't guarantee it, but it says if you chose an animal model in cancer or ARDS, these are the ones that have shown the best correlation in the past. And then the other thing that people look for is, okay, you've got a lead compound. What if something happened? Do you have a backup compound? Do you have other compounds? And the answer here is we've got over 100 of these peptides in the patents. So we've got a lot of them. So having said we've got over 100, we've only tested 14. So you may think, get on with it. But of the 14 we've tested in the lab, only 4 worked. So you may think, well, that's disappointing. No, it's actually good. If they all worked, we wouldn't have got the patent. The patent office would have said, it's obvious. So it's actually good for marks, anti-marks, that you show some work better than others, and some don't work, which is the position we're in. Doesn't mean to say the others are not useful for some other controlling mechanism, but it's not marks. Or that they couldn't be modified, put into a better formulation, look into a cyclic formulation, for example, add albumin to them. A lot of people in research do that. We've looked at a few of those through Indu, and that would be another thing we could look for in the future. But right now, we need to focus on exactly what we're doing because, okay, you affect a lot of cancers, you've got a lot of these peptides in your patent portfolio. That's terrific. But if as a small company you don't focus, then you're wasting time and money. So we are focused, obviously, on non-small cell lung cancer and on ARDS. And the reason is, I defer to Raymond. I know rhabdomyosarcoma is a very serious illness, and I'll come up to what we're doing to work with the foundation, for example, later. But in our clinical studies, these two are serious, very, very serious diseases with no really good treatment. So let's look at it first from a regulatory point of view. For respiratory, we already have an IND number issued by the FDA. In giving us the IND, that means they've reviewed all the toxicology, pharmacology work that's been submitted and accepted it. And to be fair to predecessors of me, to say toxicology was accepted, there were 17 separate studies in that toxicology packet. So this is not just trivial. There was a big packet that was accepted. We are submitting an IND update this year for ARDS, and that's not trivial either. We have to put a lot of updated data on chemistry, manufacturing, control, and I'd like to thank Indu again because he's done a lot of that work. We've got to submit all those studies, the preclinical studies that Dr. Adler was talking about, the ARDS protocol, and the investigator brochure. The investigator brochure, the FDA considers important because it's us telling the investigators how to do the study and what's all the background on the product. What about cancer in a regulatory sense? Now, this is not often the case, but this is the same peptide, remember, it's 11006. So it's the same product, but it's got a different IND number, in this case, a PIND number. And the reason is it goes to a different group, therapeutic group within the FDA. So they issue a different number for you. So this was submitted. They've accepted the protocol outline, the FDA. They didn't limit us on dose or length of dosing for cancer. And that's important because we do have use and safety data in humans. By the way, that's really unusual. When you go to the FDA about an IND meeting for a cancer drug, you've never actually put that drug into humans. So they want initial dosing to be very low and then scale up in its standard phase 1 way. Here we're going to them, and it's been in over 200 patients or subjects, and they're delighted because it's got a good safety profile, which again is not something they're used to dealing with. They're used to dealing with very toxic drugs. So again, the other important point here is they accepted cross-referencing to the other IND. In other words, we did not have to resubmit all that toxicology, pharmacology, and have it reviewed again because it's already been done. You would think that's pretty obvious, but the FDA don't always accept cross-referencing, but they did. So, we've got a product, we've got a backup product, we've got a lot of evidence it works. Which indications? There's a menu of indications, which is also good, by the way. It shows future opportunities, etc. But we could either look at reducing inflammation, and that's what the company was focused on originally, on COPD, with good rationale. It does reduce inflammation. It's not a criticism. The data from Dr. Adler's lab on cancer and ARDS wasn't around at the time. So COPD was fine. The problem is COPD is going to take thousands and thousands of patients. Although it's an unsatisfied market, there are many products around for COPD, and a lot of them are generic. It would cost us over $100 million and years of research, and we're running up to a patent expiry to do COPD. Compare that to the compelling data for lung cancer and ARDS or other cancers. These are diseases the FDA will, quote, fast-track the protocol review. You get what's called a special protocol review, and the NDA when you submit it in terms of review time. They are much smaller NDAs. You require less patients, etc. So we are focused, and this is part of the strategic change in the last year, we are focused on ARDS as part of the anti-inflammatory and lung cancer, and in particular, advanced lung cancer. And the reason it's advanced lung cancer is early lung cancer is treated by surgery or radiation, and then that's only about 10%, if that, because people are diagnosed way beyond early. So they're diagnosed usually in stage 4, much as Raymond was talking about with the children. So early lung cancers are not appropriate for this because they're treated by surgery. Stage 2 and 3 are the rage of immunotherapy. I'm sure you've read around immunotherapy. But stage 4 is they've tried everything else. You've got about 8 months to live, and there is a chemical combination, which I'll mention, that you're on. It's not very effective, and it's an awful existence for that last 8 months. So can we do something about that? I recognize money has been spent by this company on COPD, and in many ways, I'm delighted to inherit this. It's excellent data. The studies were done well. They've been submitted to the FDA, and we've got good data for safety and tolerance, particularly at the doses we're going to use. So as I said, we've done dose ranging, as you would expect to. These are the standard studies. But the outcome of that is that these studies were by no means a waste. We now have good safety data and tolerance data and know a lot more about what happens in humans when we give this drug. Now, these studies were 3 weeks long in the design of the phase 2. We're talking about giving this for 6 months in the cancer case, only 4 weeks in the ARDS, because you were either dead at the end of 4 weeks or you're alive. So that's very basic. But in cancer, we're giving it for 6 months. The FDA didn't limit us, didn't even ask for any more toxicology data. So when I went there, well, I was speaking with them, I was concerned that they may say, gee, you're taking something that you've got data on for 3 weeks and you're going out to 6 months. The reason they don't is you're treating something that's really serious, advanced lung cancer, and there's nothing else. So they didn't. So we're not limited on that. And part of that support, though, is from the COPD studies. So we are going to do studies in non-small cell lung cancer, as I keep saying. It's advanced, stage 3 or 4. I mentioned they're not suitable for surgery or radiotherapy or immunotherapy. The chemotherapy combination, in this case, it's pemetrexed and carboplatin. That is one of the most common combinations used in America for this patient population. And that's partly why I was asking all these cancer specialists, clinical oncologists, because if we do this study in, say, 12 centers in the United States, they've all got to use the same combination. We can't have the variability of them using a different combination. So this is the most likely and often used combination. I'm also keen on having one of the drugs, in this case, the second drug, doesn't really matter, carboplatin. If you remember, carboplatin is the sister drug, if you like, of cisplatin. And if you remember for Ken Adler's slide, the symbiotic or the potentiating effect of our drug and cisplatin together. So I wanted one of those combinations to be a platin because we had such encouraging data. Now, those two drugs alone are really not very effective, and they're really toxic. And even with that therapy, these patients have 8 to 12 months to live. So what are we going to do? We're going to add the peptide to the combination. Well, how do you judge whether it's working? Well, the FDA, not just related to ours, but to any cancer drug, is looking for a 20% delta, what's called a 20% delta. That's the difference you're making. So in terms of patient longevity, if you only look at that element, that's about 3 months. So can we take these patients out to an average of 15 months, for example? Improving quality of life is becoming more and more important, and it's called patient-related outcomes, or PRO. And the FDA is taking a lot more attention to this. The reason is these patients, as I said, have an awful last 8 months of their life. If you can do something, in addition to extending their life, but make their quality of life better, then that's terrific. And potentially, you could get approved on that alone, even if you didn't extend the life, if you improve the quality. So what makes us think this stuff will improve the quality of life? Well, Ken didn't mention, probably because it was one of the slides I took out, in his studies, he not only looks at the mice and their activity and videotapes them, et cetera, but he weighs them. And the mice untreated with lung cancer lose 30% of their body weight in 3 weeks, which is what you'd expect, actually. The treated ones didn't lose any weight. Now, they didn't gain weight either, but the point was they didn't lose any weight, and it was consistent. So let's say we look, does that translate to humans? I don't know. I used to treat patients, and it's really sad to see a 200-pound man go down to a 60-pound shell of what he was. That's called cachexia, but basically it's the nausea from the disease and from these treatments. So if you can hold that weight, as it did in animals, that alone would be a big improvement in quality of life. Then remember, this product was originally being looked at for COPD. Well, patients with lung cancer, I'm afraid, often smoke. And people who smoke have COPD. So the bottom line is these patients are not only going to have lung cancer, they've probably got COPD as well. And we're giving them a drug, in this case, to treat the lung cancer, but remember, it was being developed for COPD. So there's a fair chance, and we're certainly going to measure, can we improve their COPD symptoms as well? The other thing that's important for a patient, I've mentioned this in passing before, but these two drugs are injectables, and they're giving in the typically 4 cycles. So you get the injections in the hospital, you wait 3 weeks and go back again and wait 3 weeks. And the reason you're waiting 3 weeks is they're so toxic you can't tolerate another one within a week. So what's the beauty, in a sense, of a nebulizer is the patient doesn't have to go back into hospital. And it's not just the cost, but it's the inconvenience. I mean, remember, you're sick at this stage. You're really sick. So being able to do this at home is really important if we're adding a therapy. But it's unusual for a cancer therapy to be given by nebulizer. So remember, I've taken all this advice. The study design for the lung cancer study, they have to have proven advanced non-small cell lung cancer. Proven means histologically, but that's standard. All patients get standard of care, which is the pemetrexed and the carboplatin. It's a randomized controlled clinical study. So there are 3 arms, standard of care alone in this case, and then 2 different doses of the drug. These are the same 2 doses that we used in the COPD study. We know 75 milligram twice a day is well tolerated. There is some concern that there would be some tolerance issues with the 125 twice a day over a long period, like 6 months. But I'm talking about what is relatively trivial stuff compared to what a cancer drug would do to them. So irritation or cough is what I'm talking about, not somebody's hair falling out, which is what the other drugs do. As I said, the standard of care is given in 4 cycles over 3 months. Our drug is given twice daily for 6 months, and then we follow up every 3 months. So the first 3 months, well, you have a CAT scan at the beginning, and what they're looking for there is, where's your big tumor? What's the size of the tumor? How many tumors do you have? How far is it spread? So where are the organs and how many do you have? Do you have 20 metastases? Right. So then you go through the treatment regimen. And every 3 months, you have a repeat CT scan of the chest with measurements. So the beauty of this design is after the initial 6 months, it's an open, what's called an open study design. So we can look at that data every 3 months afterwards. In a normal study, you have to wait for everybody to finish the whole 12, 15 months before we can look at the data. In this design, which is an adaptive design that the FDA is recommending, you can look that data every 3 months and not get what's called a statistical penalty. And in the optimistic world, if you're showing an effect by reducing the number of metastases, reducing the size of the tumor, I mean beyond the standard of care alone, right? Then we can pick it up. Now, also, if, say, the higher dose of 11006 was a tolerance issue, you're allowed to switch them to the other dose. So you don't actually just lose the patient. These patients are expensive to enroll. So that would be a bonus. You just switch them over. You also do alkaline phosphatase, but that's standard. And if that starts an enzyme, if that starts going up, it means you've got bone metastases, and that's more likely. So imagine in the perfect world, it goes up in the standard of care group and doesn't go up in our. That would be nice. Well, it's not nice for the patient, but if we can hold. So it's worth looking at that. And then, as I mentioned before, weight and quality of life. It's important to look at as many endpoints as you can, because in these sorts of studies, you don't want to miss an endpoint where you could have shown an effect, but you didn't look for it. So there are so many elements in clinical study design. You have to give the product every chance of success. Now, you've got to run the study properly and all that sort of stuff as well. But in the design, you've got to give whatever it is you're testing every single chance you can for success. All right, I'm going to change over now to acute respiratory distress syndrome. As Ken mentioned, this is almost a kiss of death if you develop this, and very quick. 30 to 60% of people die within 4 weeks of getting this. And the reason it's such a big swing is it's very variable as to where you're being treated. I hate to say it, but in countries outside the US, that could be as high as 70%. In big centers, like University of California in the United States, where Mathey is, it's down at 30%. If you're unfortunate to live in another city in the United States, it may be up in 50%. So they're all being treated by ventilator because that's the only treatment available. But there's a big debate about is it what's called high volume ventilation or low volume? That means if you've got a patient ventilated through the mouth, are you pushing oxygen in forcefully, that's high volume, or gently, that's low volume? And logically, you'd think, well, they can't breathe, they've got problems, let's just shove it in. No, actually, you get much better results with low volume because the high volume damages the alveoli, the very end of the lung ways. So the reason I'm mentioning that again is when we do this study, we've got to standardize. Imagine we're using 6 centers here throughout the United States. We need to know, will they all use low volume? So it's not just, oh, they're on a mechanical ventilator. Yeah, but what are you doing to them? And it's all got to be the same, because if we find a difference, I don't want another company or the FDA say, well, you had a difference because half of them are doing high volume and half were doing low. So you've got to standardize and get rid of all that noise so that when you are doing your study and you find a difference, then you're very confident that it's actually because of the drug. So again, the primary endpoint here is very straightforward. How many patients, if 50% die, and say, on average, in 4 weeks, if you can make a 20% difference, 30% difference, and they don't, or less do, then that'd be huge. Because there is no other treatment. Many other drugs are being tried, like steroids, for example, have been tried in over the years and not shown to be a difference. So compassionate use. This is where we start now. What can we do? We're focused, and I think we should be focused, on those 2 indications. They're both very serious indications. We've got a lot of data from Dr. Hatter's lab to say we can genuinely make a difference. But what can we do to help the Raymond and his daughter and of this world? And the way we can do that is through what's called compassionate use. And in particular, we want to do compassionate use in children. Because compassionate use just means an investigator, in this case, the treating physician. So let's imagine, it doesn't have to be Miami, but let's imagine it was Miami Children's Hospital, now Nicholas Children's Hospital. A physician there was treating a patient and wanted access to your drug to give that patient a chance. They open what's called an investigator IND, so they refer to your IND that you've filed, but it's under their control. It gives them a lot more flexibility in dosing and things like that. And it's done quite often. When we make clinical trial supplies, which we are right now, by the way, we make, and not just us, anybody, makes significantly more than you need for the studies. And the reason is it's like a production run, right? It's more cost efficient to do that. So we plan to have more drug than we need for the studies we're doing, and we've got good stability data on the product, so we can hold it some time. And we can when a physician contacts us, we can send it to the physician to treat the patient. And obviously, not only we wouldn't charge, we can't charge. And under FDA guidelines, we can't charge, but we wouldn't charge anyway. So the patient, in this case, a rhabdomyosarcoma patient, gets a chance. We get the data. So in essence, we also benefit because let's imagine there were 12 patients treated. Without having to spend a whole lot of money to find out, we would have some indication. It's certainly not definitive. Like, I'll take two extremes. 0 out of those 12 children had any effect at all, despite those cell data. Or another way of looking at it is, well, 4 of those 12 children really did have an effect, and maybe the others didn't because of the dosing that they were given. So we could start looking at that data. So using this compassionate use gives the children a chance. It actually provides us important information and explores whether that data that was coming out of Dr. Adler's lab really does translate to humans. So it's not only Live Like Bella. I don't want to pretend that any of these nonprofit foundations we would support if it was for children. All right, so I'll stop here just for a moment, and then I'll go on again, because I want to see if there's any questions about what I was just talking about. There's one at the front first.
Right. Back on the cancer, it seems like as you're talking about there's low toxicity, but you're addressing seemingly focused for the IND on severe cases, stage 4, right? Is that to expedite through the FDA and because that's opportunistic there, or it seems like it would work at level 1, stage 1 as well, but maybe not be as easy to move through the FDA process? Is that accurate?
Right. Just for the video, I'm going to repeat his question. He's basically asking why are we focused in stage 4 rather than looking in stage 1?
Particularly given the low toxicity.
Right, given especially the low toxicity. I'd love to do stage 1. All right, I really would. This drug, if it works, has huge potential. I said stage 1 is treated by surgery, and it is. But they do not have metastases. They've got a primary. They were lucky. They were diagnosed early enough. By the way, this is only 10% of the market, because 90% stage 4. But there is a huge opportunity because now you're talking about prophylaxis. What if you gave this product in the intention of preventing them ever getting secondaries? Why not that study? Because
Could you just put it in a cigarette?
that'd be pretty ironic. It's one way of inhaling it. The problem with prophylaxis rather than treatment studies, treatment studies, what we're planning, okay? They have a definitive end to them. Prophylaxis studies are longitudinal studies, and you've got to follow these patients out 10, 12 years. So it's a huge cost. Now, if Pfizer had this product, they may look at it that way. But no, we're going to stay with treatment.
Dr. Dixon, with these several tests that you plan, how far does the current supply of 11006 take us into that, particularly with it being dose dependent as well?
The clinical trial supplies, we've got over $1 million, this will come up later on, but we've got over $1 million worth of active pharmaceutical ingredient. That means API, in other words. So we've got over $1 million of that, and we're using nearly all of it to have made into clinical trial supplies. That means they've got to be put into the formula to what's called good manufacturing practice with the FDA. we have made, or that batch would make enough for us to do the ARDS study and the cancer study, and that'd be it. So if we wanted to go on and do phase 3 cancer studies, for example, we would have to make more API. Does that answer your question? Right. But
Couple questions. How long will the two studies be? The cancer and the ARDS? And then the second question is there any possibility for a priority review voucher, or I don't think it fits in that category, but how about a pediatric one?
do I need to repeat those questions? I can hear them. I'm just asking for the video. All right, for the recording. There was 2 questions there, at least. One was in terms of would we get a priority review or a voucher? The answer is I believe we would. It's possible we'd get one for both. If we didn't get one for non-small cell lung cancer, it's because there are so many studies in that area. The ARDS, I believe we would, yes, because it's such a severe disease. The other question related, although you didn't use the term inflection point, but I think that's where you were going, was how long will it take these studies to be completed? And the answer is we hope to start these studies in the last quarter of this year, both of them. And that is driven by the time taken to make the clinical trial supplies and then submit the IND updates. We can't really submit the IND updates even though we've got them pretty much ready because we need the manufacturing data from the clinical trial supply manufacturer to add into that CMC section. So the real answer then is once we start, then enrollment is the rate limiting factor, all right? Because ARDS, you're going to be limited as to which centers, and the centers have got to be really committed because the patients come in acute distress and need something immediately. So you've got to be right on top of that so they remember, the clinical nurse remembers, oh, let's put them in the study. Because they're under some stress. They've got a patient dying and needing to go on a ventilator. So the beauty of the ARDS study, though, is, yes, enrollment is going to be not easy, but the actual study is very short. It's only 4 weeks long. Or, we will follow them up, by the way, but the prime endpoint would be at 4 weeks, 6 weeks. So I would hope to see data next year, towards the end of next year, for ARDS, and know whether we've got good chance or not. The lung cancer study would be probably the following year because we've got to follow them up for 12 to 15 months. The only upside there, an upside, would be, remember, we're getting data every 3 months as we follow them out. So if we got really good data and we didn't need to go for 12 months to show an effect, then we'd know earlier and potentially it would be earlier.
I think I heard on BBC radio last night that Prince had gone into the hospital with serious pneumonia. Is it by chance, would he have been an ARDS potentially patient?
I didn't hear of a diagnosis other than he had, quote, a respiratory disease. Was all I heard.
Speculation.
Yeah, could be. I don't know. At the back?
I have 2 questions. Question 1, who's the treating physician for ARDS? Pulmonologist? Question number 2, I've seen some incidents in the transplantation of renal and lung. Have you looked at that population? Obviously, it's very high unmet need, multiple issues in that patient population. Starting point, that's it.
Right. So again, for the video, I can hear you. But, For the video, the question was, who is the treating physician usually for an ARDS patient? And the answer is the director or the head of respiratory critical care. So on those slides I was showing earlier, for example, at UNC, that's Dr. Shannon Carson. So it's a physician, it's not a pulmonologist. Pulmonologists do long-term care, not acute critical care. So it's the head of the respiratory critical care unit. And then your other question related to other forms of acute lung injury. ARDS is one form. But for example, lung transplant rejection, could you make a difference there? The answer is potentially yes. And ironically, just 2 weeks ago, Ken and Indu applied to the NIH for a grant just for that. So we've just applied for a grant, it's preclinical, obviously, to see if we can prevent acute lung injury or lung rejection. So, yeah, it's potential. Yeah, sorry.
If you get good data in the ARDS trial, what would be the next step? Would you go right to a phase 3 trial, or would you still have to do an intermediate stage?
It depends how persuasive the data is. If it's marginal, then you finish your study and you look and discuss with another meeting with the FDA whether what size of a study you would need to do in phase 2 or 3.
If that's the case,
Right.
getting
All right.
good data,
I just hesitate on the best case, all right? The best case would be if you did show a difference and it wasn't marginal, right? It's a big difference. The FDA would probably ask you to keep going with that same study to build up the number of patients so that the data is more compelling, right? So you've got two options. One, if it's marginal data, they're likely to tell you, finish your study, let's talk about it, and look at the design element of a follow-up study. But if you've got really good data, there has been instances where they'll just ask you to keep enrolling in the same protocol. Tom.
Let me just comment that if the data is really good, somebody will come along and say, can we buy this? Or can we?
Well, they'd be wanting to buy us, not this. Well, it could be this, but
Exactly.
much more likely to be us. Yeah. Yeah.
Question. We haven't seen a lot of information about Biomarck in the press. Are we getting to a point where we might begin to see things in the press about these developments?
Yes, I'd like to think so. And you're absolutely right. There's been very little information about Biomarck in the press. So we're looking at how we could improve that. And there are many avenues to do that. And I don't want to say they're officially in the lead, but Laura Calamos, who's sitting there, is on the board. And she was looking at even getting NPR to come to this meeting. So we're looking at those sorts of connections. And Tom Roberg is connected to the state congressman and senator. Well, some of them, the ones he fights with. Well, you ran against the congressman. I don't know if that's a great friendship, but anyway, the point is, yes, we are looking to increase our PR in general. At the back. Well, there's always a possibility, but you run out of product. And some companies hate doing compassionate care studies. And the reason is it puts you into a position of, do you supply or not? And frankly, it's a position I'd like not to be in. So if we say we're doing compassionate care, I think we have to give some boundary. You know, we're going to be sending out drug all over the place. And the boundary is, in what I'm suggesting, would be children and certain disease states. The reason is, let's imagine it was rhabdomyosarcoma and neuroblastoma, just those two. Why limit it to those two? Because we've got preclinical data that it does work in those two, so there's a rationale. There's certainly a need, all right? And I'm getting more data. I'd rather get 12 patients worth of data from rhabdomyosarcoma than one patient from 54 other cancers, because I can't do anything with it. I can't do much. I'm not pretending, by the way, that data from a compassionate use program is much other than interesting. And it's certainly not persuasive. Yeah.
In your clinical study for cancer, you're going to do the 75 milligrams versus the 125 milligrams.
Correct.
How do you know, or can you test that 500 would be the best number? Is it just the statistical difference between 75 and 125?
The reason we're not, again, the question is why not go to a higher dose in cancer? And it's a very rational question, and I thought the FDA may push us. Remember, we've shown a dose response, in other words, in animals. So the higher you go, the better result you get. And certainly that's true. But we've also done COPD studies. And remember, those doses were being tested. There was different doses, once a day, twice a day, higher doses. And 75 twice a day was, I'd say, well tolerated. But you started seeing symptoms at 125. And above that, we had symptoms. So we know already what the upper limit should be. Yeah.
Any potential for orphan drug designation for subpopulation?
Certainly not for lung cancer. It's so common. ARDS is marginal. If you remember, the incidence that Dr. Adler was quoting was 200,000. Well, it's now 500,000 or so. So you're bumping along right under the orphan drug designation. But yes, for ARDS, there's a chance currently. Depends how many firefighters or how many wars we fight and people get blast injuries. Because that's where this disease was first noticed. It's actually in Vietnam. Any other questions? Yeah.
Comment was made that given highly significant trial results, the company might be purchased. What is it that will be something that Pfizer will want? Is it the knowledge of the interaction between the Mark and the 11006? Is it the isolation work that you're doing? Is it the interaction? What part of that is able to be protected by that?
The question is, what would interest a big company to either co-develop with us or take us over or buy it, acquire it? Right. What they want is clinical data. Right. So there are, I can't say who it is, but there are 5 companies awaiting that we've already spoken to in the last year that are wanting to see, have asked to see the clinical data when we have it. So that's what's going to push them over the edge or not. And again, if it's marginal clinical data, maybe not. If it's impressive, yeah. Now, they won't just look at that. In doing their due diligence, they'll be looking at the method of action data that Ken was talking about and all the other things that I've been talking about in terms of dose relationship. All of which, and your patent protection, your portfolio, and all of that sort of stuff. But the one thing that would push them over the edge, and they're waiting for, is clinical data. So we, as a company, have to get clinical data. I will tell you, I do not believe we, as a company, will end up taking any of these indications through to an NDA. Because if we get as far as doing a phase 3 study, you're likely to be acquired. That's not a bad thing. It means the drug would still be developed, maybe many more indications, and they'll take up rhabdomyosarcoma, for example, as well. And you people, well, as a CEO, I report to the board, and the board's job is you as shareholders to maximize the value. That's what we're doing. So it is likely that the endpoint for the shareholders or exit would be an acquisition or a buyout. It could be a co-development partner too.
It's my understanding that Vice President Biden has been put in charge of some type of looking at cancer. Has anybody from Biomarck or Raymond been in contact with Vice President Biden? If not, I think what I heard from Dr. Adler is that he was saying at this presentation, nobody's heard this before. So if I was Vice President Biden and I heard Dr. Adler today, I'd be very interested in putting some money into Biomarck.
Right. The question is, has anybody from the company spoken to Biden and his team, because he set up a team to look into cancer cure? And the short answer is yes. Tom Roberg on our board has been in contact through congressional staff and Senate staff. I don't know if you remember, but a few months ago, Biden was actually here in Raleigh. Right. Yeah, right. And Tom Roberg and I were invited. So we do some publicity. So we went to the round table with him. Oh, sorry.
I also met with Vice President Biden's advisor, who's heading up the meetings for the Cancer Moonshot in Capitol Hill last month. I delivered them the research documents that we have from Ken Adler's lab and talked to them, and I'll be meeting with them again next month, actually in 2 weeks from now. So we have multiple members of us interested in both the governmental approach to being involved in moving cancer research forward. And I know that Raymond is very involved in his efforts as well through the White House. So we're trying every approach.
All right, just to repeat that, because I think some of the people at the back probably couldn't hear it, and I'm sure the video. In addition to, this is approaching Biden or his staff related to what we're doing. In addition to what Tom is doing, Laura Calamos is also on the board, is in Washington frequently on other business, and has met with Biden's staff and given them part of the presentation. So certainly, Laura has been active and will be active in that.
Given the success in the clinical trials, what would be a likely model of monetization with the pharma company in terms of like cash, or equity, or royalty, or other?
It depends what a deal is. A typical deal structure for a license. So say they wanted to license 006 for cancer. There would typically be an upfront, and then stage payments, and then royalty. The upfront depends on how far you've got and how compelling your data is. So the more compelling, the further you've got, say to phase 2, for example, more upfront. The least data you have, the less upfront. The stage payments relate to end of phase 3, for example. You've done your definitive studies, that would be a stage payment. NDA approval typically is a stage payment. And then the royalty would be single to low double figures, royalty, percentage-wise.
Well, this is a horrible question to ask, but big pharma is big pharma. What would be the chance that they would want to buy us and then never develop the drug because they have their own drug that they think for billions on?
It's a possibility, and normally you can put clauses in an agreement to say that you can't not develop it. They're not lock-all, though, because, they could develop it slowly, should we say. So even though you've got all these protective clauses in, I wouldn't want to pretend that that's the all I know. It's unlikely they'd bother to do that, though. Certainly not with ARDS. But cancer is a possibility as you go through. But we try and protect against that because obviously we want the thing to be developed. We'd have to get approval from the board anyway to do a deal like that. Yeah.
I'm German, so please excuse my English. Would it be possible to sell both fields to different companies, especially as it seems to me that the RADS situation will come to a much faster result than the lung cancer thing. So could it be sold to two different companies at two different times?
Yeah, the question is, could you sell two different, more than that, indications separately? So take cancer and ARDS to two different companies. The chance of that is not very high because it's the same drug. In this case, Bio 11006. So a company would buy it for whichever they want and may or may not develop it for the other one. In these two indications, they probably would, because your price point is going to be about the same. So, in other words, if you got this to market and you look at other drugs similar to this, like cancer drugs are on the market, they're priced pretty high. So if you look at an ARDS drug, it could also be priced pretty high. So you've got a similar price point or likely price point for this. Whereas, this is not a criticism of COPD, but for COPD you couldn't do that because your price point is going to be so much lower. So but for these two indications, the price point would be about the same. So it could be put on the market as the same. The other option is to take the second of our peptides. Remember, we've got four that work, 10901, and develop that for whatever you want to develop for, ARDS, and bring that out as a separate drug. And in a sense it is, and in a sense it isn't. Because 11006 is the 10 amino acid peptide. 10901 is a 9. It's the same, it's just one shorter. So, and when your body takes in 11006, within minutes, that amino acid is cleaved off and it becomes 10901 in the body. So, the regulatory authorities may have a difference of opinion about whether those are two different drugs or not. So that's a long way to say. Can I go on, or should I assume you're not interested in an investment opportunity? I do appreciate the questions, though, and that's part of the value of having a meeting like this, so I've got no issue with that. Okay, as I'm sure you're aware, we intend to raise $6 million at a company valuation of $120 million. So where did that come from? Well, we looked at published completed is important, all right? Completed deals for the 18 months prior to August last year for companies in a similar position to ours. In other words, well, we looked at preclinical or phase 1. We could argue we're in phase 2, but let's just stay with that. In other words, we didn't look at completed deals of companies that are in phase 2, phase 3, because they would be huge. And then we discounted the valuation that we got as an average valuation by 70%. That's not because we were being nice, it's because we don't have a completed deal. So a completed deal is obviously worth a lot more than a non-completed deal, so we discounted it. And what are we going to use the funds for? Well, it's for the clinical studies. I'm just pointing out we don't need it for operating costs. We can cover that ourselves. Another way, it's not really a way of valuing a company, but one way, especially for a private company, is so, what's happened in the last year, or what's happened in the previous 3 years? Well, let me take the last year. And by last year, I mean April last year to April this year. So it's what's happened in the last 12 months? Well, the answer is a lot. Clearly, we've done a refocus onto cancer and ARDS, and it's pretty obvious why. We've had the pre-IND meeting with the FDA for cancer, and it was a good meeting. Remember, they didn't limit us on how long to dose it, what dose, all that sort of stuff. We have an NIH grant for COPD, and they agreed to switch it to ARDS. That's important because it's a $1.1 million grant from the NIH. A company database was established. You may think, so what? It's really important to have all your data in one place. So it sounds trivial, but it was a lot of work, driven by Alistair, who drives everybody nuts, but he was right to get it done. The data room has been established. That's important too. What a data room is say Pfizer come to us and say, we'd like to see your cancer data. In these days, you have an electronic data room. You don't send them bucket loads of paper, but you have to have it designated and blocked off on your file, and it has to have all of the cancer data, in this case, organized by a logical flow, and it's got to be complete. And then you give them a password to get into the data room. So we've established data rooms for both cancer and ARDS, so that when companies come and knocking, we're ready for them. The beauty from that also is we can tell who took access and what they looked at, but that's electronic surveillance these days. So data room is not trivial. We keep prosecuting our worldwide patent and for cancer and for ARDS, for that matter. And it's pretty obvious that research has continued in MOA, the method of action, in both cancer and ARDS, because a lot of that was what Ken was talking about. So what about this year? For the first 4 months, what's gone on? Well, as I said, we're doing these IND updates for both cancer and ARDS. It is a lot of work doing it. The CMC, the protocol, the investigators, just the protocol, as you can see, there's so many moving parts to a protocol, and you've got to get it right. The clinical trial supplies are being prepared, as I mentioned, so we're using that API now to make those clinical trial supplies. We've had quotes, so we know how much we need in money sense to do our studies. So contract research organizations, 5 of them have quoted. There's 2 that I would look at again. The cancer patent issued. I don't want to trivialize this in any sense. This is only 2 weeks ago, and Indu Parikh has been leading the charge on this, but that is huge. So our patent, we were notified, our cancer patent, we were notified 2 weeks ago, has been approved by the patent office. That gives us patent extension out to 2034. That would certainly interest an acquiring company, because that's a huge amount of time. And not only was it approved, but I don't know how well comfortable you are with patent laws and stuff like that, but companies typically put in a lot of claims, because the patent office typically thins them back hugely. We got most of our claims approved. So it wasn't just that we got the cancer patent approved, it was the breadth of the claims we got approved that, frankly, I was delightfully surprised with. So, good work Indu and the team. And then the company website has been updated. We took it down at the end of for the last 6 months of last year because it was so out of date. We released it again in January this year. It is always going to be a work in progress, but it's going to be updated several times a year. We are about to release one next week, hopefully on Monday. So I would encourage you to look at the website periodically and just see what changes we've made. So what about the financial position? The company currently has $1.4 million in cash. So that's where we stand right now. We've got that grant that I mentioned from the National Institute of Health, for 1.1. That has to be done for odds, but that doesn't matter. We can use other money for NCL. The company has over $1 million worth of API, as I already mentioned, to make the supplies. And we have investor commitments, current investor commitments, over time, and they are paid and are current. With a further, it's actually 3.0. Oh, that's me. I put 3.1, but it is 3.0 million due. Half of it on the first patient enrolled into a cancer study, and half of it on the last patient enrolled. So we're operating on a minimal overhead. So we don't have a huge structure, which is okay for now, but we do need to hire 2 clinical project managers to run these studies properly. So again, the purpose of this new funding is to enable us to increase the size of these studies, because we've got a much better chance of showing success by increasing the size and therefore the power of the study. If it's a really small study, then you could miss a positive result. So, I mean, you can go overboard too and do one too big. But we're on the low end, put it that way. So that's why I would suggest and have suggested that we raise money and increase the patient numbers we can enroll. So in any of these investment opportunities, we go through risks. Clearly, although there's compelling data in animals, and they're the most predictive models for humans, the results can't be guaranteed in humans. You know, it's pretty obvious, but we have to state it. And results from the clinical trials can be definitive, suggestive, or negative. And again, we don't know which one of those. The reason we want to increase the number of patients, as I just said, is to increase the likelihood of a definitive or persuasive study. What about the reward? Well, we've talked a little bit about this, but the first 2 bullets relate to a financial reward, which I'm sure you're all interested in. So a positive result in either cancer or ARDS would substantially change the value of the company pretty much overnight. As soon as you know those results, the value changes hugely. You know, as I said, we go to companies that we know and have relationships now with. So the value, don't need to work. If that was the case, you wouldn't be looking for a long exit. It'll be pretty quick. The estimated annual sales, for example, and there are many examples of this, of a drug for non-small cell lung cancer would be over $1 billion a year. And there are already drugs on the market that are easily that. And for an ARDS drug would be over $800 million a year. So these are very attractive markets financially. But then the last 2 bullets, you know, it's not all of life is about money. So these are both very serious diseases. And any positive result would change lives. There's no doubt about it, whether you're a cancer patient dying an awful death, or you're an ARDS patient trying to be one of the lucky ones to make it through that 4 weeks. So it would certainly change the life. These are serious, serious diseases. And one way of looking at this is every 15,000 we raise, one more patient could be entered into these studies, which obviously says they're about $15,000 a patient, which is the hospital treating costs plus the monitoring costs. So not only would you be adding a patient to the patient database to help us have more definitive data, but hopefully you'd be helping change the life of whichever patient that was as well. So there's a serious outcome on both ends. So I've finished, you may be pleased to know, and how about that for timing? It's 12 o'clock on the nose. So thanks for participating. Does anybody have any questions about that section? Well, I couldn't answer that, to be honest, because I'd love to be able to answer that when we have forward sales. But right now, you're looking at a valuation model based on income. And
the projected sales of companies that you've researched was $800 million to $1 billion.
right, what I was looking at there was not their valuation. The valuation related to companies in our position, not in same. The 800 million or the billion were companies who had already launched, in the case of cancer drugs, or are looking, they've done an IPO, as there was a company that is looking for ARDS work. And in doing their IPO, this was in London, by the way, in January of this year, that was the investment bank's estimate of the market. So that's where that came from.
It seems that the stuff's awfully expensive to produce, and do you take into account on the $800 billion in your valuation whether or not these other companies have that kind of cost in producing their product?
Well, peptides can be expensive to produce, there's no doubt about it. But like many other things, that's related to the volume. So it depends how much you're trying to produce. If you're doing a low volume for clinical trial supplies, you're not getting anybody excited. They want to make commercial peptides. Insulin, for example, is a peptide, just to say there are peptides on the market. Well, if you've got high volume, your prices are going to come down. But right now, for us doing clinical trial supplies, you know, we need with kind of 50,000 vials, that sort of stuff. They'd be interested if you were ordering 500 million vials, but at 50,000, they make you pay more for it.
That's because of economies of scale and just producing it.
Yes, they've got to take their line down from potentially making a commercial product and make your research product and then clean it all to GMP and they'll get inspected. So, you know, they say, oh, whoa, we've got a lot of overheads tied up here. And they do, to be fair to them.
There's one more point, if I can address on the peptide. I've been to the Sorry.
Have it.
Do you mind?
Yeah.
I've been to the manufacturing
This
site.
is what chairmen do.
Sorry. Myself, 3 times. There's a patent that we haven't filed yet, and it is the process of making this peptide. So we have time to do that, but when the time is right, we can file this patent, and then that'll give us another extension on just the API itself.
Well, you might as well close if you're up there.
If there's anything else, I'd be happy. I want to thank everyone again, especially, you know, our friend Raymond. I hope you enjoyed today's present. Oh, I'm sorry. Please.
Yeah, has there any consideration now given whether they got Adler or some other, looking at 1106 applied to other cancers?
Yes. Yes, yes. So the question is, have we looked at 1106 for other cancers? The answer is yes. If you recall, there was a brief presentation about triple-negative breast cancer. What the team is doing, Brian especially, is we're trying to enter into this clinical phase, which is towards the end of this year, and then it gives a little breathing time. And during that clinical trial, which is going to be run by a CRO, we'll have time to work with Ken's lab, do other studies, hopefully do something with Raymond as well, and generate more, because there could be another home run out there that we talk about on a regular basis. So the answer is yes. Yes, please.
The election forms are due next Friday, is that correct? For current investors?
Correct.
Okay, and, I thought I saw a closing of May 6th. Is that funded, or is the funding gonna be later than that?
No, that's for the new investors. It will be a closing of that date. So if you're a current investor, it is sooner than if you're a new investor. It'll be the later date, May 6th.
I'm sorry. May 6th
What's it- is that the date,
current
right?
investor
And it's 30 days, right? 30 days after that is
Yes.
the later? Yeah, so 30 days after that will be the new investors.
But the election form is kind of I emailed Brian about it, a range of what I might commit. But then a week later, you want check and subscription agreement. Is that what I'm hearing?
Correct. Is that what the email was?
Yes. If you're a current investor and you want to invest pro rata to where you are now, then there is a minimum related to how many shares you hold. So it will vary for everybody. You know, if you own 50,000, there'll be a certain minimum to stay pro rata. If you own 100,000, be certain. But we're trying to give priority to our current investors in time. And then the new investors can add. And you do not, by the way, obviously, have to put in just your minimum. You could increase. All we're saying is the pro rata minimum would be this. And if you want to check, you could check. Is Rob Futrell still here? Oh, there you are, Rob. I don't want to swamp you with 100 people calling you to say exactly where we are. I mean, I was assuming that people knew how many shares they had if they were a current investor. That may be a blind assumption. But
Feel free, they can contact us and we can provide that information.
all right. I would suggest if you do contact Rob Futrell, he's from Wyrick Robbins, and he did that Manila document set, to email him. I would ask you not to phone him because it's gonna rack up the company bills. So I'll be very blunt about it. Please email him, and he'll have somebody email you back and answer your question. All right. I guess now I'm going to close as we did a change. Again, I'd like to thank you all for coming. We intend holding future meetings in this venue, so because we've been happy with it. I'd like to thank the technical support back there, who really did help us, and hope to see you all again in the future. The documents in the Manila folder are the financial documents. The others are the copy of the slides, obviously. I'd ask you to be careful. I mean, if somebody else is interested, you're welcome to hand them out. But as Ken mentioned, they do have some data that hasn't even been published yet. Publish it, Ken. Anyway, it has been patented, so I'm not that nervous. It's been included into our patents. All right, thank you again.